Hello everybody and welcome to today's webinar. On behalf of European Pharmaceutical Review and GenScript ProBio, I'd like to thank you all for attending. I'm your moderator, Caroline Peachy, the editor of European Pharmaceutical Review. Today's speaker will be Dr. Lauren Liu, the Senior Director of the QC Department at GenScript ProBio. Dr. Liu has more than 10 years of experience in analytical methods development for biomacromolecules, including recombinant proteins and antibodies, plasmids, peptides, and viruses. During today's webinar, Lauren will be discussing the diverse applications of plasmid DNA products in the market, including the latest guidelines from the European Medicines Agency and the US Food and Drug Administration. And she'll be discussing both the commercial applications and clinical trial materials. So after the presentation, we'll be moving on to a live question and answer session. So please remember you can submit your questions at any point during the webinar using the questions panel on the menu in the right. So thank you for coming. I'm delighted to hand you over to today's speaker now, Dr. Lauren Liu. Lauren, over to you.
Thank you. Okay, so thank you for joining today's webinar. So today's topic is about the challenges in analytical strategy for plasmid DNA projects. And yeah, sorry, I want to try it work. Yeah, okay. So in today's talk, I'm going to talk about the 4 parts. So firstly, the first part is about the current plasmid DNA products in the market. So as with the rapid growth of the gene and cell therapy area, we can find like there come out so many different advanced therapy medicinal projects, that is ATMP. And here I just list like 4 major categories of the gene and cell therapies that is very popular used in this GCT area. So the first category is the cell therapy. The cell therapy is very well known that I just take the CAR T therapy as example. CAR T therapy is very well known that it's really a very amazing therapy that they can cure people. So generally speaking, this therapy is firstly, it works like this way. Firstly, we track the cells for patients. Then we need to use an antivirus to edit these cells, and then make this cell to express some specific antibodies to make this cell become a weapon that when to inject back to the human being, these weapons can kill the blood cancer cells, and in this way, the patient can be cured. So antivirus in this cell therapy is very, very important starting material that we can see here. We normally we need to use like 4 plasmid systems to pack the lentivirus. So here we can see like 2 kinds of plasmids here. First is the helper plasmid that will like to express some proteins to the frame of this virus. And then it's another kind of plasmid, transfer plasmids. The transfer plasmid can deliver the gene of interest to the lentivirus, and finally it will be add into the CAR T cell and become make the CAR T cell can really work in the human beings. And here is the first is cell therapy, and next can we move to the gene cell therapy. In gene cell therapy, I take the AAV therapy as an example. So usually AAV can be used to deliver therapeutic genes into the cells or tissues to change some mutant or damaged genes, and so that this therapy can cure genetic disease. And for the AAV, if I want to take AAV, we usually use like a 3 plasmid system. So that's very similar to the lentivirus. They have 2 kinds of plasmids here. First is the helper plasmid that can be to express some the protein of AAV, and another is target protein that can deliver the gene of interest to the AAV. That finally this delivered gene will be transferred to the human beings. And the next is the third categories of the gene and cell therapy. It's the gene vaccines. Gene vaccine actually it's like it's defined. Vaccines are defined as purified plasmid mixtures that contain maybe one DNA or more DNA sequences that capable to induce the immune response in human being that can protect us from a pathogen. And here we can see here, so in this DNA vaccine, usually we can contain the plasmids, and to make this drug can work, vaccine can work. So this is the third category, and last category is the mRNA vaccine. So mRNA vaccine, it's I think due to the epidemic called COVID-19 virus disease. So I think everyone everyone knows mRNA vaccine is a very powerful vaccine that can prevent us from the pathogens or virus. And for this mRNA vaccine, like it's also like to first they need like a template plasmid DNA, and then the plasmid DNA can transfer to into I mean to like into use this gene a plasmid vaccine to like produce mRNA, and this mRNA can also teach our body how to make some very specific protein that can help us from some disease or some viruses. So, so in this cases, this all this the key gene information that can be expressed finally in the human beings come from this template plasmids. So so when we look at these 4 categories, gene and cell therapy, I think it's very easy to find that the plasmid was everywhere. They're everywhere, and it's really plasmid. It really work a lot and act as a different roles in our cell and gene therapies. So so since plasmids can have so many different roles here, so let's to check. Yeah, so I'd like to introduce some regulations to will help us to identify the roles of this plasmid PN projects in this so many different applications. So here I collect like 7 major guidelines downloaded from the EMA or the FDA. So I think it's very interesting to find that can find here that most of these guidelines like just issued in recent 4 years, and also we can find another is a lot of the guidelines just to give some guidance for the IND stage, and yeah. So this is it's obviously tell us that the cell gene therapy is a brand new area all the industry are trying to get a very good practice to meet the regulatory requirements and to make our cell and gene therapy areas requirements. And so when we establish our ProBio's QC actually we refer a lot of principles and strategies to these regulations, and this is so when if we want to try to identify the different roles of these plasmids in all these various therapy applications. So I think it's very important as to really understand these regulations want to tell. So in order to better understand our today's presentation, I would like to introduce some terminology here. It's about two terms. It's starting material versus drug substance. Yeah, as I mentioned before, there are so many various therapy scenarios, or when we're using the plasmid DNA, and actually if we want to really to disclose these roles, we first need to learn this term. So regarding the starting material, from Q7 and also the EMA's guidelines, there's some very good interpretation of the starting material. It is a raw material intermediate or an API that is used in production of an API and that is incorporated as a significant structural fragment into the structure of API. And it's just come from the Q7. I think the one guideline from the EMA is very well to describe the this definition in the ATMP. So these starting materials are defined as all the materials from which the active substance is manufactured or extracted. So it means for some plasmid DNA products, even though it will not be directly injected into the human beings, but it can deliver the final gene of interest to the human being. So all these plasmids can be treated as a starting material. And the next term is raw material. Raw material actually in Q7 it's a very general term to use to denote like starting material or reagents or solvents intended for use in the production of intermediates or API. And in EMA's guideline, actually, I think it's a more precisely description. So the raw materials used in the manufacture of active substance are not intended to form part of active substance. So how to understand this is, for example, the help plasmids of the lentivirus. Actually, when this lentivirus is used in the CAR T therapy scenario, this helper plasmid can be treated as the raw material, not just such a starting material, because this helper plasmid cannot deliver some gene of interest to the final product. It's only like responsible for the service, so some proteins of this lentivirus service. Yeah. So next is the I need to clarify it's the drug substance or the active pharmaceutical ingredients API. And in the Q1A, ICH-Q1A, it said like any substance or mixture of substance intended to be used in the manufacturing of drug product, and that when used in the production of drug becomes an API. Yeah, so usually when we in our biological area, this we usually call it like drug substance. It means very active pharmaceutical aid ingredients just in our drugs that will be directly injected to the human beings. And for the drug product, it's more easily to understand. It's just the final dosage form that will be intended using on marketing or directly inject to the human beings. So after we can really understand in gene and cell therapy, so for the ATMP, what does anti-material means and what does drug substance means, I think it's better understand the roles of plasmid DNA in all these kinds of applications. So I just like summarize all these situations and to see what is the position of the DNA plasmids from the very first beginning. I think it's very important to understand the plasmid DNA's role. So I'd like to divide all these applications into three parts. First is ex vivo gene therapy, and the next is in vivo gene therapy, and finally the vaccine. Because it's three totally different therapy scenario, and will use different guidelines. And in the ex vivo gene therapy, we will it usually contains some like cell therapy, for example, CAR T therapy, TCR-T therapy, or some cell modification. So we can see here. So the for lentivirus, we got like transfer plasmid, helper plasmid, and for some like ex vivo gene transfer, we also use some. We usually have got some non-viral vectors, and for the Cas9 mRNA used to modify the cell, we also have some template plasmids that will be used to generate a Cas9 mRNA. So in all the situations, the help plasmid in lentivirus and in the template plasmid that will be to produce Cas9 mRNA, we can treat them as like raw material. And other transfer plasmids of non-viral vectors, we can treat them as a starting material. And the second part is the in vivo gene therapy. So it's like this therapy will be directly injected into the human beings. It's included in vivo CAR T or AAV injection. I just leave these two application here. So for all this, for example, linearized DNA that we use to produce mRNA, or the transfer plasmids or helper plasmid to use the AAV, since all the mRNA and AAV will be direct inject to the human beings. So all this linearized DNA, transfer plasmids, and helper plasmids, we treat all of them as a starting material. And finally, in the vaccine, we got the mRNA vaccine and DNA vaccine. So for the linearized DNA, the role is quite like the help plasmids or AAV. It's also treated as a starting material. But in a DNA vaccine, because all this plasmid DNA will be part of this active ingredients that will be formed in the final drug product. So we treat all this plasmid DNA as a DS or DP. And when we're looking this all starting material and raw materials, so how can we normally to using some practice? So according to the EMA's guideline recently, so all this starting material or raw material, when we manufacturing them, we usually follow like GMP principles, not its current GMP. We try to using principles to to control very critical quality attributes of this starting material or raw material. But for DSDP, we must to follow the current GMP to do the manufacturing. And since the regulatory is very key for us, so when we do the submission either for the IND or for the BLA, we usually use the ECTD document. So I also summarize here is if this plasmid DNA products can be treated as a starting material or raw material, actually we can use very simplified CMC information summary to do the submission. But for the DSODP, we just using the normal CMC dosages. Yeah, just like normal monoclonal antibody DSDP. Yeah. So finally, I just talking about this ECTD document since it's a very critical document that is related to the submission. So I just like summary here is just as acid. We this plasmid genes can be treated like raw material, starting material, DS, and DP. So actually in the CTD module, they will be listed in different chapters. And for example, for the raw material, starting material, we usually like put them in the control of the materials this chapter. Yeah, but in the DSODP, usually in the module 3, the DS part or the DP part. And yeah, this is this part is about our current plasmid gene products, and we can see here they have different applications, and according to the regulatory requirements, we can define them either as starting material or either as a DSODP. And then I move to next part. It's about analytical CMC challenges. So this slide is my favorite slide. Actually, the quality by design is principle. I think is very popular in our area, not just mentioned, yeah, in monoclonal antibody or just in GCT area. So here I just need to mention two keys here: process and product. No matter it's DSDP or a starting material, when we want to develop a process that process can result in a very good control quality product, we need to know the process and product. The whole CMC development actually is a gradual understanding of the molecule and the process, and finally leading to the implementation of the appropriate control strategy based on risk assessment. So this due to the QBD concept, so actually in the whole CMC development, not just for starting material, also about DSDP, we can see actually they have so many analytical activities here. So in the front part, here I get the plasmid stimulates the whole give the whole CMC process stages of the plasmid DNA. And so we're starting from the Lexicon construction and the cell bank and fermentation, purification, formulation, and finally it's power to manufacturing. So during all this process development, we got so many activities, analytical activities, where during the process development, we need to establish security, we need to develop methods, and we even need to do some identify some key substance here. And when we move into the pilot manufacturing, is more like GMP practice, we also contain so many works here, either specification or master verification, validation, or do some release testing, still have some stability studies. All these activities will be it concludes in the whole plasmid development. And all this process actually, I think it's the consistent with the QPD concept. All this quality analytical work of plasmid DNA during the CMC development is the process of gradually understanding the unique molecule and process. So since we talk about this two key elements here is the molecule and the process. So in order to identify more differences in the starting material or the DSDP for the plasmid products, I like to first to introduce what's the differences between the starting material and the DSDP in terms of both molecules and the process. So I like to summarize in the 6 aspects. So first is the molecule structure. So when plasmids DNA used as a starting material, actually they can be used in different area, lenti, AV, or mRNA. So it will result in such situation is the plasmid structure of this kinds of during this application, actually we got some very special unique structures. For example, the ATR region for some plasmids using AAV, and we also got like poly A tail structure for some plasmids that will be used in mRNA, and also the copy number of this plasmid actually is vary a lot. And if we see the plasmids for the DSDP, really we can find that for the copy number is really like a moderate, not so varied a lot. And for the whole structure, it's also not so many special or special structure inside the plasmid, but we usually pay more attention to the code optimization. We're trying to get a very enhanced yield. And for the microbial cell bank, actually is using a starting material. We can start from the PCB or MCB, and all the stability of this cell bank will be focusing on the yield change. But in the DSDP, for DSDP, so we usually need to like build PCB, MCB, or WCB. And as for the stability of this microbial cell bank, not just including the yield change, we also need to pay attention to the genetic stability. And for the fermentation and downstream process, actually, as for starting material, we need to like just to make sure the batch consistency. And also when we do the purification, actually we can balance yield and impurity levels, because when you use as the starting material, impurity removal level totally depend on the end of use. So you don't need like a very you don't need require very nice. So this is the major difference between the fermentation and downstream process. And I like to address is the formulation buffer. So for a starting material, usually we're using like fixed platform buffer. But for DSDP, according to the dosage form, we need to we need to do some optimization of formulation. And finally, it will result like the buffer, the final formulation buffer will be different for each other. Yeah. And lastly is about the pulse scale. So when the starting material, when plasmid is used as a starting material, actually this pulse scale is usually like less than 50 liters, usually like makes like several liters. Yeah, especially during the IND stage. But for the DSDP, we usually like find that this, or even on IND stage, we might also require like a more than 200 liter scale. So yeah, when we know this different features of plasmid molecule and the process between the starting material and this DP role, then we can easily to get some conclusions here that what analytical challenges for the plasmid DNA products. Actually, all this essential, all this why this challenge can occur is due to because the plasmid virus got very diverse applications. So in terms of method strategy, diverse application may lead to different securities, and the diverse application might lead to a different method life cycle management strategy. And second part is the certification, also because of these diverse applications. So different control level for the securities, it will be, yeah, it must be different control levels. And the last one is the characterization strategy challenge. And I think this all challenges belongs to all the biological molecules, because biological medicines really it's a very large molecules. It's a very complex. Really, we cannot use one method to tell the one quality result, one quality attributes. We need the orthogonal methods trying to will identify one quality attribute. And then in next part, I'm going to like to give more case study that how ProBio to meet these difficult challenges. How can we use current practice to meet these challenges? So first case is about the method life cycle management. So actually in ProBio, actually we propose a very first propose like a project specific qualification strategy for our method. So during the IND stage for the plasmids that will be treated as the starting material, we suggest use the PSQ strategy to do the method qualification. So there are three facts here is in our platform, we already using our platform and analytical procedures to finish like huge CMC huge plasmid CMC projects, and all this that we already done its method validation for our platform analytical procedures. And also in our QC lab, we are using a GMP Pro or GMP quality system, so we can 100% sure about the data integrity. So once we use this PSQ strategy, actually we can use in very scientific based experiments and can meet the requirements for IND or EMA, FDA, and MPA guidelines in very efficient ways. Also, we can guarantee the data integrity. So this idea is come from this analytical procedure lifetime cycle concept. So this figure we like just summarize from the ICHQ 14 and USP 12 and 20. So during the method development, actually all this method is starting with the analytical target profile, and according to the analytical target profile, we are going to select one method to develop method, qualify method, to validate method, and then we also need to continue to verify our method until it can be as a long-term use long-term QC use. But but as plasmid used as a starting material, actually we need to notice that first, all these plasmids are very similar to each other, and second, all this plasmid buffer is already like a fixed platform buffer. And also for the IND stage, we just need to do some, we can just meet the IND stage because all the methods are defined to meet the fit for purpose. And according to these three facts, so we propose this PSQ strategy. And I just give example here about some methods in terms of impurity or the identification test. So usually for the DSDP, when we to codify a method, we need to like do the specificity, precision, accuracy, linearity, limit of quantification range, or robustness. But as for a starting material of the plasmid DNA product, we can use profiles or platform method procedures only just finish the specificity and accuracy qualification, and then we can directly use our platform method to release test our plasmid DNA products. Also for the identification test, yeah, so we can to like to just using just to qualify specificity, and then we can directly use our platform method to release this plasmid DNA as a starting material. Yeah, this is the one case here. And the second case is about the specification setting strategy. Specification is very critical, not for the quality analytics of plasmid, not not only not easy, even though no matter it is starting material or DSDP. And this specification usually contains the three parts: the quality attributes, test methods, and criteria. But this how to set the specification actually is like a lot of factors will impact this setting. The molecule structure can impact methods, impact impurity can impact, yeah, and also all the process related, the process understanding, the manufacturing control, or batch to batch consistency also can impact this specification setting strategy. As I mentioned before, the difference between the starting material and DSDP plasmid DNA products is that for the starting material, we only use like platform process. We're using platform methods, and when we develop process, actually we more pay attention to the batch consistency, or pay much more attention to some critical quality attributes. So when we set the specification for the plasmid products, we need to realize the difference between these two roles and to get appropriate certification for the different use. And then I just give an example in our platform here is when plasmid DNA is a starting material or as drug product. Actually, we can see here, I use some red words to emphasize the difference between these two different roles. So when a plasmid is used as a drug product, actually, we in terms of general properties, we need to pay attention to some non-visible particles or some insoluble particles. But as for the starting material, I think this will be not critical quality attributes. And also about there's another quality attribute is the DNA homogeneity. It's very important for the DNA for the plasmid DNA. And actually, according to our plasma experience, So when it is as a starting material, usually we can the requirement for this quality attribute usually like just larger, not less larger than 80%. But when we move to the use of the drug product, actually we can see that this subcoiled purity actually is much more higher than that is used in starting material. And also there's another different part is about impurities, just I mentioned before. So the purification process actually is different between these two rows, and that is a result into here the difference. So for these impurities, actually when we use the starting material, this control level is quite not so strict because there when it treated as starting material, it means there still have some downstream process where it becomes impurity. So usually this impurity will be controlled not so much. But for the DS and DP, you can see here in our platform, it's very strict, have very strict impurity level controls. And the last one is about potency. About potency, so for the drug product, it's very important to develop potency even at the R&D stage because this potency assay can really reflect the efficacy in the clinical trials. But for the starting material, actually this potency is really will not be part of this critical quality attribute. Yeah, and next case is about the characterization of these toposomers. And for the supercoiled form, actually it's very well known that it's a very critical quality attribute for the plasmid DNA. And we know there is like a supercoiled form or like open circle form. But actually, supercoiled form also have different various morphology, and we can see in the left figure here. So depending on the different tightness of the twisting, so actually the supercoil form can perform different topoisomers. And in the right part is about the two different orthogonal methods that we can well define this different can differentiate the supercoil and dimer or open circle. Yeah, and actually we found a very interesting story here is when we using the HPLC, the normal peak is like a symmetric chromatography peak, but sometimes we can find a very abnormal peak, just like a cliff peak from HPLC. But what really this cliff peak is, we we design some experiments. We try to using the capillary geoelectrophoresis to like orthogonal analysis this influence. So we collect influence from the chromatography and we using the CGE to analysis. So we very interesting that we found that all this influence in the in the electrophoresis, it shows that all of them are supercoil. And then we do use some enzyme to make this influence change into the linear form or the open circle form. And also we found in the in the electrophoresis gram, we found that all this just one peak. It means all this study means that the HPLC can separate different supercoil forms. Yeah, although it looks like the cliff peak, but actually all of them are supercoiled form. And another case is about powerful sequencing technology. So so as I mentioned before, ITR sequence, ITR area is a very special area. Actually, they got so many high GC content, and usually it's very hard for either the first generation or next generation sequencing to do. And in our platform, we try to using like single molecule sequencing. We combine the single sequencing NGS and the single molecule sequencing. We finally can really to identify to sequencing this ITR area to identify each nucleic acid. So last case is about the poly characterization. As I mentioned, when the plasmids use a different area, so actually they got different quality attributes. So poly tail is a very typical structure for um, the plasmids that we use in the mRNA. So usually we also like two different methods to like orthogonal analysis of this poly tail. But we're using central sequencing, but central sequencing they can sequence the poly tail, but it will usually like limited to like 120 NT. And also they have a disadvantage, they cannot able to quantify poly tail distribution. And we also use the CE. CE is actually a very good like quantitative method to will quantify the poly tail distribution even up to 150 NT. And here we can see the case here is we're using the sync sequencing and the CE to first to identify to categorize poly A tail in the plasmid DNA. We found that this poly A is like at least like 128, and the distribution might the distribution is range from like 116 to 157. And then once they using this plasmid to transfer into the mRNA, we found using this result is quite consistent with the mRNA product. We can see in the right part is the mRNA result. The poly distribution we are using the LCMS to identify this mRNA result is ranging from 122 to 131, and also we can see the LCMS photography here is the only very pure one peak. It means this poly A tail is quite the percentage of the poly A tail is quite high. So strict control of poly A tail of plasmid DNA can really guarantee the mRNA's poly purity and distribution. This part, I just need to spend like 3 minutes to introduce our high standard QC platform in ProBio. Actually, we established this QC lab in the 2018, and we actually got kept in pace of the latest guidance. And actually, we also participated in drafting domestic regulatory guidance in CGT area. And the whole QC lab is under operation. All the operation is compliant with the EU, US, and GMT, and already passed so many audits without any critical findings. And for the methods, as you can see from my case study, we got very state-of-the-art plasmid analytical form, not only for release testing, but also for characterization. And for equipment, in our QC lab, we have internal team to do the CSV and do the instrument qualification. And here is the facilities construction plan for our consistent support. Our business platform really can support from preclinical to commercial. And we can see here, till now, we already got like one plasmid BD and GMP Pro building, and we got another plasmid GMP building. We have like one analytical development lab and one full functional QC lab. And in next 2 years, we are going to expand to GMP facility, and we're going to expand to QC lab in China. And also in US, we'll be launch our GMP plasmid facility and the full functional QC lab in 2024. Last, I want to just to share some our excellent track records and for our plasmid platform. Actually, we've already obtained more than 20 IND approvals globally, and now there are 60 IND projects ongoing, and we already finished more than 140 GMP probe batches for clinical use. And in our platform, we have a licensed strain with with licensed authority, and it's for the lentiviruses hyperplasma, we have the CMF registered at FDA and a very clean history and IP free, and it's also royalty free. So in the right figure, it's just the more detailed information about our IND approvals. It's you can see here, not just FDA, we also got from the Netherlands, from the Japan, from the Korea, and from Australia. Yeah. So although not just so many years, but our plasmid platform really got some accomplishments here. So yeah, thank you.
Fantastic. Thank you, Lauren, for a really interesting presentation. We're going to move on to a short question and answer session, but before that, we've got a few short polling questions for you. So the first question: have you ever heard of GenScript ProBio or GenScript? Please, can you select one of the options? So only GenScript ProBio, only GenScript, both GenScript ProBio and GenScript, or have you heard of neither? Please, can you respond now? And just a reminder that you are able to submit any questions you've got for Lauren in the box on the right-hand side. Right, we'll just keep this open. So this question: have you heard of GenScript ProBio or GenScript? Please, can you select one of the options now? Okay, second polling question: which services are you interested in? So we've got plasmid manufacturing, we've got lentivirus manufacturing, AAV manufacturing, CMC services for plasmids, LVVs or AAVs. Please, could you vote now? Thank you. Just leave this open for just another couple of minutes. And then shortly we'll be going to the live Q&A session. Okay, another question. When do you need services? Is it within the next half a year, within half a year to 1 year, within the next 1 to 2 years, or are you looking ahead more than 2 years? I'd be really grateful if you could select an option. Fantastic, we're getting some votes in. And as I've said, we've got one more question and then we'll be moving to the Q&A session. So please submit your questions as well on the right-hand side. Okay, and one final question, and this is relating to a topic for a future webinar. What topics are you most interested in? Is it cell and gene therapy, mRNA, DNA vaccines and drugs, antibody discovery, or antibody and protein contract development and manufacturing, or other? Please respond. Amazing. Okay, fantastic. Thank you all for those responses. I'm now delighted to introduce our question and answer session. So don't forget, you can submit your questions, and any that we can't get through will be responded to after the webinar. Right, so for the first question, we've had an audience question in. So let's go to that one. The first question, Lauren, for you. In a DNA vaccine, or in fact any other DNA medicine, is it normal to have the product controlled with a certain amount of double-stranded DNA?
Yeah, so my gut question is means to control the double strand DNA. Sorry, the last sentence is when in a DNA vaccine.
Yeah, so in a DNA vaccine, is it normal to have the product controlled with a certain amount of double-stranded DNA? And it says this would contribute to the potency of the drug product.
For the I just want to clarify if this double strand DNA means the linear DNA. So actually, is that for the DNA when that's plasmid DNA used as the DSDP? Actually, when either you using the chromatography or using the electrophoresis, you can see many peaks here. So in our study, actually, if we using this plasmid as DS and DP product, we need to identify all these peaks. What are they? Usually for this so this plasmid forms really not just like a supercoiled form, but open cell co-core form. We also found that during the process, usually you can find like double strand DNA, like linear DNA. Yeah, it did exist, and you need to identify things because all this DNA cannot work. So it can kind of like impurity. So yeah, my answer is yes. You need to find them. You need to identify them. Yeah.
Fantastic, thanks, Lauren. And we've got another question, and it relates specifically to slide 21. And it asks, is there a reference or guidance for impurities exceptions criteria for DP? And it says, to a second part of the question, it says, for example, host cell protein to be controlled at less than or equal to 0.1.
For when the plasmid DNA used as a DP, all this I think is totally depend on your end use. How say it means when you use as a DP, you need to know your MOA of your drug, and you need to know the dosage of your drug. So when your dosage is very high, it means you need to control the host cell protein level. And if you really want to find how to define this level, I think I'm not sure the detailed chapter number. So in USP chapter, I think they give some guidelines like you need like control the dosage of this protein because it will all this host protein can result in some safety issues. I think this is one chapter in USP chapter, and in EP I'm not sure if there is so detailed information here. And for the for the residue DNA, actually, I remember in the ICH guideline there is a control level about at least you need to control how much of this residue hosts the DNA. And in the recent FDA's guidance for the guidelines for the DNA drug, I remember there is also a number show you the limit, the minimum level you need to control. Yeah.
Okay, fantastic, Lauren. Unfortunately, that's all we've got time for today. But any additional questions, if you have any questions, they'll be responded to via email. So I'd like everyone to join me in thanking Dr. Lauren Liu for her time today. And as you leave the webinar, a survey will appear on the screen asking you to rate the session. So please take a moment to provide your feedback. And if now's not a good time, we'll send you an email shortly via email. If you could complete it, we'd be greatly appreciated. So on behalf of European Pharmaceutical Review, GenScript ProBio, I'd like to thank you all for attending today's webinar and hope to see you soon. Goodbye.
Bye.
Bye. Thanks, Lauren.